Now, this is the diagram which shows the whole process of excitation contraction coupling. Okay, so in this diagram we can see many things. Okay, first of all, uh, let me tell you that this is the myocardial cell. Okay, this all is myocardial cell, and this is the sarcolemma or the cell membrane, this one. Okay, and you see the invagination of these sarcolemma, which are known as transverse tubules. Okay, and you see each and every transverse tubule has got two terminal cisterns, okay, on either side, and this is known as the triad. Sarcoplasmic reticulum has got high concentration of calcium inside here. So the calcium is stored in the sarcoplasmic reticulum, and you know that the calcium is actively pumped from the sarcoplasm, from the cytosol into the sarcoplasm, and is stored there. And there is a protein present there in the sarcoplasmic reticulum, which is known as the calsequestrin, okay, which actually binds the calcium ions and and the calcium is stored in the sarcoplasmic reticulum. Here you can see sodium potassium pump, which is the primary active transport. Here you can see calcium sodium exchanger, which is basically secondary active transport. Okay, so and this is the calcium ATPase in the sarcoplasmic reticulum membrane, and this ATPase is also known as SERCA, if you remember, the sarcoplasmic endoplasmic calcium ATPase, SERCA, sarcoplasmic reticulum calcium ATPase. And then you can see these are the voltage gated calcium channels in the sarcolemma, voltage gated calcium channels, which are basically L type of calcium channels. Then you can see channels in the membranes of the sarcoplasmic reticulum, and these are the calcium channels. Okay, calcium channels in the, and these channels are also known as ryanodine receptors. Okay, these are the same channels which are present in the skeletal muscle fibers. Okay, ryanodine channels. So what happens that the action potential moves around or travels around the sarcolemma, and it goes deeper into the transverse tubules. Okay, and when it reaches here near the sarcoplasmic reticular terminal cisterns, over here it causes opening of the voltage gated calcium channels. During the phase 2, the plateau phase, okay, the voltage gated L type calcium channels are open, and the calcium ions, they go inside the myocardial muscle fibers. Okay, and this calcium enters the cells, and this calcium which has gone into the cells causes the release of the calcium from the sarcoplasmic reticulum, and this is known as calcium induced calcium release. These channels, ryanodine receptor, are open when the calcium enters the myocardial cells. So this is known as the calcium induced calcium release. So the calcium level inside the myocardial fibers, the calcium concentration, calcium ion concentration rises. Okay, and this increased level of calcium leads to binding of the calcium ions to troponin C, and that exposes the active sites on the actin filaments, and to which the heads of the myosin, they get attached, and through sliding filament mechanism, that process of contraction occurs. So you can say that it is the calcium ion which enters from the outside. The extracellular calcium ions actually triggers the release of the calcium ions from the sarcoplasmic reticulum, and then the calcium level within the myocardial cell rises, which binds with the troponin C and causes the process of contraction. Okay, now when the contraction is over, what happens that this calcium which was released is pumped back by this calcium ATPase, which is also known as SERCA, back into the sarcoplasmic reticulum. Plus calcium is also moving out of the myocardial cells in exchange for sodium, which is a secondary active transport, and then energy to this transport, secondary active transport, is provided by the sodium potassium pump. You know, the sodium potassium pump pumps the sodium out, okay, in exchange for potassium. So what happens that the sodium concentration is greater outside and it is less inside, and this gradient for the sodium ions actually provides the force with which the calcium is pumped out. Okay, the calcium is pumped out against the concentration gradient. So the calcium is moving uphill against the while sodium is moving down the concentration. So this is an secondary active transport, and this is because of this. This secondary active transport is taking place because of this primary active. So in fact, what happens that the calcium is pumped back into the sarcoplasmic reticulum, or it is pumped outside, and as a result of that, the calcium level within the myocardial fiber, cardiac muscle fiber, it decreases, and that results in the process of relaxation. Now on the left side, this whole process is described in the form of a flow chart. So first of all, the cardiac action potential arrives, and as a result of that, during the play 2 phase, the calcium enters the cell, the cardiac myocyte, and then it causes calcium induced calcium release from the sarcoplasmic reticulum. Calcium induced calcium release from the sarcoplasmic reticulum, and then this calcium binds to the troponin C, and that leads to the cross bridge cycling, and that results in the contraction. Okay, and during the relaxation, what happens that the calcium is pumped back into the sarcoplasmic reticulum by calcium ATPase, and it is also pumped out in exchange for sodium. Okay, so the calcium level in the myocardial cell falls, which results in relaxation. So this is the whole process of excitation contraction coupling. Now if you compare it with the skeletal muscle fibers, remember that in skeletal muscle fibers, these L-type calcium channels, they do not allow the calcium to go inside. They are not opened. Okay, they are not there. So the extracellular calcium ions does not play any role in the process of contraction. But in the heart, the force of contraction or the contraction depends a lot on the extracellular calcium ions. So extracellular calcium ions plays a very important role in the process of contraction, in the initiation of the contraction in the cardiac muscle fibers. But in case of skeletal muscles, the extracellular calcium ion has no role in the initiation of the contraction process. In the skeletal muscles, the process of contraction is initiated by the calcium which comes from the entirely from the sarcoplasmic reticulum, and the extracellular calcium ions has no role in it. While in case of cardiac muscle cells, the contraction depends on not only on the calcium that is coming from the sarcoplasmic reticulum, but also on the calcium that is coming from the outside, that is the extracellular calcium ions.